So hi everyone, welcome to the EViNS webinar series hosted by the EV Club. Thanks to Ken. I am Christian Neri, the chair of EViNS, and today we have the pleasure of having Mitali Tyagi from the lab of Jason Shepard. So Jason Shepard was the chair together with Rosemary was the chair of the second conference on EVs in nervous system a few weeks ago in Montreal, and that was really a great success. And we have the pleasure today to listen again to the work of Jason on Alzheimer's disease. So, Jason, thanks to be here today with Mitali, and I leave it to you to introduce Mitali for the talk she's going to give.
Thanks, Christian, and thanks for Ken for organizing, and thanks everyone for joining in. So Mitali is a senior student in my lab. She's actually just about to graduate and leave the lab, unfortunately, and start a postdoc in David Holtzman's lab. But before she leaves, we're also just about to resubmit her paper, and so she'll talk about her data. And we've been, sort of stumbling into the EV field partly because of this discovery that we made a few years ago about some genes that encode proteins that can make capsids. And this Alzheimer's idea was really allowed by a grant that I got from the Chan Zuckerberg Initiative that allowed us to take some risks, and it's sort of taken a life of its own, and we're excited for you to see this and welcome feedback. Yeah, Mitali, you can take it away.
Thank you for that introduction, and thank you so much for having me today. I'm so excited to share my work that we have been working on for the past few years now. All right, yeah, so today I want to share the story about how we figured out that ARC mediates intercellular tau transmission through extracellular vesicles during Alzheimer's disease. As we all know, Alzheimer's disease is a very debilitating neurodegenerative disorder, which is accompanied by a lot of hallmarks, such as tau aggregation, A-beta aggregation, vascular alterations, neuroinflammation, synapse loss, neuronal loss. But the two major histopathological hallmarks of Alzheimer's disease are tau aggregation and amyloid beta aggregation. So if we were to look into the brain of an Alzheimer's patient post-mortem, what we would see is that hyperphosphorylated tau aggregates inside the neurons and makes these neurofibrillary tangles. And amyloid beta protein aggregates outside the neurons to make these amyloid plaques. And though both amyloid and tau pathology are extremely important to understand Alzheimer's disease, what we have seen is that tau pathology correlates very strongly with the symptoms that you see in Alzheimer's patients. To give you an example, right here, I have a baseline amyloid PET from an Alzheimer's patient, a baseline tau PET from the same patient, and a brain scan showing brain atrophy from the same patient. And I hope you can appreciate that the areas that have developed tau pathology coincide perfectly with the areas that are undergoing brain atrophy. So this shows that somehow tau pathology could be driving the symptoms that we see in Alzheimer's patients. Another interesting thing about tau pathology during Alzheimer's disease is that it starts in one area of the brain, but it spreads all over the brain eventually in a very stereotypical pattern. So what you would see in an Alzheimer's patient is that tau pathology starts in the locus ceruleus or medial entorhinal cortical area. And over the period of time, it will spread to hippocampal regions and then to neocortical areas. If we were to look deeper into how the tau pathology spreading all over the brain, we see that tau makes monomers, which then makes protofibrils and then mature aggregates. These mature aggregates can then fragment into little seeds. And when these tau seeds are transferred between neurons, it causes cells to cell spreading. And when they are transferred between two brain regions, it causes region to region spreading. So the big question that we are trying to address in our lab is, how does the intercellular spread of pathological tau occur? And what we know so far is that if you have a neuron that has developed tau pathology in it, it would have tau fibrils. And these tau fibrils break down to make little tau seeds. These tau seeds are then released from this neuron. And when these tau seeds are taken up by a healthy neuron, the tau seed comes in contact with physiological tau in this healthy neuron and somehow corrupts the physiological tau there, starting tau pathology in a healthy neuron all over again. And it is known that these tau seeds can either be released in their naked form or they can be released in extracellular vesicles. We call this vesicular tau. The Ikizu lab at Mayo Clinic a few years back showed that this vesicular tau is much more potent at spreading and seeding tau pathology. They basically got some post-mortem tissue from Alzheimer's patients, and then they isolated either EV tau from them or oligomeric tau or fibrillar tau. And they injected equal amounts of these different forms of tau in, I think, 12 to 18-month-old mice in their DG area. And then they basically tracked tau seeding and spreading in these mice. And you can see over here, the mice that were injected with EV tau basically started developing tau pathology in them. You could see tau positive cells, and it was also spreading. But if we look at the mice that were injected with oligomeric tau or fibrillar tau, which both are forms of naked tau, there was not much tau seeding happening. And that is quantified right here. So this shows that EV tau is much more potent at tau seeding, and maybe that is something that we should be focusing on. And so that got us wondering, how is tau getting into extracellular vesicles, and how could it be spreading tau pathology through extracellular vesicles? Our lab works on this really cool protein called ARC. It stands for Activity Regulated Cytoskeletal Associated Protein. It's an immediate early gene that is rapidly transcribed in response to neuronal activity. To show you this, I have some in situ hybridization right here. This is the hippocampal area of a mouse. And you can see at basal levels, there is not a lot of ARC mRNA in here. But if you give high frequency stimulation to induce neuronal activity, it just blazes with ARC mRNA. ARC is also known to be a master regulator of synaptic plasticity and memory consolidation. It has also been observed that the rats and mice that do not have ARC, they develop impaired long-term memory. To give you an example, this data was collected by a talented grad student within the lab, Alicia. So basically, she took some wild type and ARC knockout rats and then did some fear conditioning on them. You basically take these rats and put them in a fear conditioning chamber. And then during training, we give them a series of foot shocks in a certain context. And then after 24 hours, we test their recall by putting them in the same chamber and seeing if they would freeze. If they remember that this was the context where they received the foot shocks, they basically would freeze out of fear. And when she did that, she saw that the wild types did freeze quite a bit, remembering the context in which they received the foot shocks, but the ARC knockouts could not do that. Our lab a few years back made this very exciting discovery that ARC also makes these capsid structures, these virus-like particles. Over here, I have some HIV capsids, and down here, I have some ARC capsids. And if you compare the two, it's really hard to differentiate between the two. And it is very astonishing why a neuronal gene would be actually making capsids and what role could that be playing. And our lab figured out that ARC makes these capsids and also has a novel form of intercellular communication between neurons, which is basically facilitated by these ARC capsids. So how it works is that if you have a donor neuron, what you could see is that there are ARC monomers in there, and these ARC monomers then make ARC capsids. These ARC capsids are then packaged in extracellular vesicles and released from the donor neurons. When these ARC-containing extracellular vesicles are taken up, the ARC capsids are released in a recipient neuron. And it's also known that the ARC capsids can actually take RNA or protein with them. At this point of time, I hope you can appreciate that the way tau is getting transferred between neurons and ARC is getting transferred between neurons is very similar. So that's what motivated us to basically pursue this. Just to give you a little bit of more background, other grad students within the lab, Kaylin and Alicia, they basically looked into how ARC is getting released in extracellular vesicles. And they found that an IBAR protein, IRIS-P53, which can basically curve the membrane, is involved. They found out that ARC binds with IRIS-P53, and then both of them get packaged in extracellular vesicles and get released from donor neurons. And these ARC-containing extracellular vesicles can then be taken up by neighboring recipient neurons. So all of this led to the question, does ARC play a role in intercellular tau transfer? And to test this, we used a tau transfer assay that was developed by the Hyman Lab at Harvard. We basically have an AAV that encodes for EGFP2A human tau with P301L mutation. This mutation is associated with frontotemporal dementia. The idea being that you will get a monocistronic RNA that will encode for EGFP2A human tau, but you would get two different proteins, GFP and human tau, in a neuron that has been transduced with this virus. So a donor neuron would be a neuron that is expressing both GFP and tau after getting transduced with this AAV. And if we are able to detect any neurons that have tau but no GFP, then that would indicate that there has been an intercellular tau transfer event. So again, the donor cells would be both GFP and tau positive, and the recipient neurons would be tau positive but GFP negative. So firstly, I used this virus in cultured neurons. I took some wild type and ARC knockout primary hippocampal neuronal cultures and then transduced them with this virus. And after 14 days, I fixed these cells and then stained for tau, human tau specifically, and GFP. If we look at the representative images here, I was able to detect transduced donor neurons in both wild type and ARC knockout cultures. There were some intercellular tau transfer events as well, which was super exciting. If we have a closer look at this merge channel right here, what we see is that in the wild type cultures, close to the donor neuron, which is indicated by this yellow arrow, we were able to detect a lot of recipient neurons indicated by the orange arrows that were just tau positive and GFP negative. But when we look at ARC knockout cultures, the number of recipient neurons around the donor neurons is significantly reduced. So this shows that ARC somehow is involved in intercellular tau transfer. And this is quantified right here. We have done this multiple number of times in cultures, and I'm showing you data from 3 cultures right here. Next, what we wanted to check was, okay, so ARC is playing a role in intercellular tau transfer in cultured neurons, but would we be able to capture this in vivo as well? To test this, we injected the same virus in 6-month-old wild-type and ARC knockout mice in their medial entorhinal cortex, where the tau pathology starts in Alzheimer's disease. And then after 10 weeks, we collected their brains and then stained for GFP and tau again. Here's a slice just to show you our injection site and imaging site. We injected in medial entorhinal cortex right here. And for this to work, you need sparse transduction. That's why we chose to image on the contralateral side where we were getting sparse transduction. We were able to detect GFP positive neurons in both wild type and ARC knockout mice, showing that our AAV worked and the transduction was working. We were also able to detect a lot of just human tau positive neurons, showing that intercellular tau transfer was working in vivo as well. When we look at the merge channel right here, you can see that in wild type mice, around donor neurons indicated by yellow arrow, we were able to detect recipient neurons that are indicated by orange arrows. And if we look at the ARC knockout mice, the number of recipient neurons around the donor neurons is significantly reduced. When I quantified the GFP intensity over area for these mice, there were no significant differences between wild type and ARC knockout mice. This shows that the amount of AAV transduction that happened between these mice were pretty comparable. But when I look at the number of recipient neuron per donor neuron, it's significantly reduced in ARC knockouts, showing that in the absence of ARC, the intercellular tau transfer decreases significantly. Next, we wondered if ARC facilitates release of tau in EVs. To test this, we generated some new mice. We got RTG4510 mice that have P301L mutation in their tau gene. And we crossed them with either wild-type mice or ARC knockout mice to generate tau transgenic wild types and tau transgenic ARC knockout mice. And we used them at 4 months of age because this is the age where they have full-blown tau pathology. They start developing it around 2.53 months, but by 4 months, you have full-blown tau pathology. So very talented previous postdoc in the lab, she developed a brain EV isolation protocol for me. So she basically took 2 mouse brains, just the forebrain area, and chopped them up and then used collagenase to break down the tissue. And then after serial centrifugation steps, she used size exclusion eyes-on columns to isolate brain EVs from these mice. She also went ahead and validated these EVs. We were able to detect positive EV markers such as ALIX, syntenin, and CD9 in these EVs right here. We did not see negative EV marker histone 3 in our preps, which was promising. And we were also able to detect ARC in our EV preps, showing that we were able to isolate EVs that do contain ARC. Right here, I have some brain homogenates just to show you that all these proteins were present in these brain homogenate samples. Next, we did some NTA to validate the size of the EVs that we were getting, and this was mostly exosomes. Our peak was round about 110, 120. So yeah, we validated our EV preps really well. And next, we wanted to know what kind of tau is getting packaged in these EVs. And so, we used a number of tau antibodies to test this. We used tau 13 and tau 22 that stained for oligomeric tau. And then we also used 88 and tau Y9 that stained for phosphorylated tau. We also used a lot of other antibodies that I'm not showing today, but the whole idea being that we were able to detect oligomeric and phosphorylated tau in these EVs, which would indicate that's the kind of tau that is getting packaged in these extracellular vesicles. We also wanted to make sure that tau was actually inside the EVs and not just associated with the EVs. So we did some proteinase K assays. So when we did not add any proteinase K or Triton X, which is a detergent, we were able to detect both tau and ARC in these EVs. Syntenin is a positive EV marker right here. When we do add proteinase K, but no detergent, the EVs should be intact, but all the protein around the EVs should be disintegrated. And we see that tau 13 and ARC is protected inside the EVs along with syntenin. But when we do add detergent, which breaks open the EVs and allows proteinase K to degrade all the other protein, tau 13 and ARC goes away. So this shows that both tau 13 and ARC are protected inside the EVs. Next, we wanted to compare the levels of tau that we were seeing in tau transgenic wild type and ARC knockout mice. So we isolated brain EVs from 4-month-old tau transgenic wild types and ARC knockouts. And right here, you can see there was a lot of tau in the EVs isolated from tau transgenic wild types, but it was almost absent in the EVs isolated from tau transgenic ARC knockouts. I also have ARC and syntenin as positive marker for these EVs. When we quantified the amount of tau that was getting packaged in the EVs, we saw that it significantly reduced in tau transgenic ARC knockouts. But when we quantified the amounts of syntenin in these EVs, there were no significant differences. So it seems like that the EV production itself is not affected, but the packaging of tau in these EVs are. We also used the same EV samples to do some tau ELISA just to confirm what we were seeing in Western blots, and we saw a similar effect in total human tau ELISA. But just to be sure, we wanted to double check that the EV production was not affected in the absence of ARC. So we isolated EVs from 4-month-old tau transgenic wild types and ARC knockouts and did some nanoparticle tracking assay on them. When we calculated the number of particles per mil, there were no significant differences between the two. The median particle size was pretty comparable, and even the peak particle size was pretty comparable between tau transgenic wild types and ARC knockouts. So this assured us that it was not the EV production, but the packaging of tau that was different. This was very cool, but at best, using Western blots, we were getting to the same EV fractions. We wanted to know if ARC and tau are physically present in the same EVs. So to do this, we did some double immunogold labeling. We used primary antibodies for ARC and tau, and then used gold particle conjugated secondary antibodies. In the 4-month-old tau transgenic wild type mice, we were able to detect EVs that just had ARC in them, EVs that had just tau in them, and then there was a special population of EVs that had both ARC and tau in them, which is shown right here. So this was very exciting for us that there are some EVs that physically package ARC and tau together. Next, we wondered if these ARC tau EVs can actually seed tau pathology, because if they can't, then they are not actually relevant to Alzheimer's disease. To test this, we used a FRET-based tau seeding assay, which was developed by the Diamond Lab. The whole idea is that we have hex cells that are expressing a tau repeat domain that does not aggregate on its own, but if you provide a tau seed to them, they can aggregate. So the tau repeat domain is conjugated to either CFP or YFP. And when we will add tau seeds, either in naked form or in EV form, the tau repeat domains would start aggregating, bringing this CFP and YFP together. That would generate a FRET signal. So I transfected these HEC biosensor cells with either tau transgenic wild-type EVs or tau transgenic ARC knockout EVs, an equal amount of these EVs. And then collected those cells and did some flow cytometry on them. Over here, you can see the HEC biosensor cells that were transfected with tau transgenic wild-type EVs did show a special population of cells that were FRET positive, indicating that there was some tau seeding in these samples. But when we look at the HEC biosensor cells that were transfected with tau transgenic ARC knockout EVs, this population is almost absent. This is quantified right here. So that shows that these tau transgenic wild-type EVs that contain ARC and tau are also capable of seeding tau pathology and could be relevant to Alzheimer's disease. Next, we wondered, how does ARC facilitate the release of tau in EVs? And to test this, we did some GST pull-down assays, wondering if ARC and tau could actually bind together. So I took some GST ARC or GST endophyllin, and then I incubated them with cell lysate collected from non-transfected, wild-type tau transfected, or mutated tau transfected hex cells. And after incubating them together, I collected the elute and ran Western blots. Here are the elutes from these different proteins. As you can see here, the GST ARC elutes contained tau in them, which I validated using two antibodies, HD7 and tau13. But the elutes collected from GST or GST endophyllin did not contain any tau in them. This shows that ARC and tau bind together. Next, I wondered if this was a direct interaction. And to test that, I took purified GST or GST ARC and incubated them with purified tau protein, and then tested the elutes for tau. And you can see right here that the elutes that were collected from GST ARC did contain tau in them, but the elutes collected from GST did not. So this shows that there is a direct interaction between ARC and tau. Next, I wondered if this interaction also exists in vivo. So to test that, I took 4-month-old tau transgenic mice, and then I immunoprecipitated ARC from their forebrain. After that, I blotted back for tau and iris P53. As I mentioned before, iris P53 is a known interactor of ARC and also helps ARC in getting released in extracellular vesicles. So in the IPs, in these ARC IPs, I was able to detect both tau and iris P53. So this shows that ARC, tau, and iris P53 could be present in a complex in these mice. Next, I wondered if ARC would have more affinity for hyperphosphorylated tau, which is a pathological kind of tau, in comparison to physiological tau, which sort of was indicated here. If you look at the mutated tau, the amount of tau that I was getting in these elutes was slightly higher than the wild type tau, but I just wanted to be sure. So to test this, I incubated GST ARC with either tau that had phosphorylation or with tau that did not have phosphorylation. I basically took phosphorylated tau and then used a phosphatase to remove all the phosphorylation sites to get non-phosphorylated tau right here. And after incubating both of them, I collected elutes like I did before, and then blotted back for tau. Over here, you can see that the elutes collected from phosphorylated tau, the amounts of tau that we detect is way more in comparison to the elutes that had non-phosphorylated tau. So this shows that ARC somehow does have more affinity for hyperphosphorylated pathological tau. And it's quantified right here. I'm showing the levels of elutes normalized to the input. Next, we wanted to see if the ARC tau EVs could affect tau pathology in these tau transgenic mice. So we took 4-month-old tau transgenic wild type and ARC knockout mice. And then after collecting their brains, we just stained for total tau in them. On top, here is the panel of tau transgenic wild type mice. And down below here is tau transgenic ARC knockout mice. If we look at the magnified images right here, I hope you can appreciate that in tau transgenic wild type mice, it seems like the tau pathology is way lower in comparison to tau transgenic ARC knockouts. If you look at the neurons right here in the CA1 region in tau transgenic ARC knockouts, it almost seems like tau is getting stuck in the cell bodies of these neurons. When I quantified average tau expression per neuron in these mice, I saw that it was significantly increased in tau transgenic ARC knockouts. I also observed something really interesting when looking at these mice. I saw that the cell body layer in the CA1 region of these mice, basically in tau transgenic ARC knockouts, there seemed to be some thinning of this cell body layer, which could indicate that there is neuronal loss happening. So I went ahead and calculated the total number of new and positive neurons in these cell body layers. And I saw that it was significantly reduced in tau transgenic ARC knockouts. So this could indicate that if you knock out ARC, you basically tau cannot get into EVs and then could be accumulating in the cell body layer right here in the neurons. And that could cause cell toxicity and lead to cell death in these neurons that cannot release tau in EVs. All of this was very exciting, but again, there are caveats to using transgenic mouse models. It's an overexpression system. So we wanted to see if we can recapitulate all of this in humans, which technically is the only non-overexpression system. So we started with the question, does ARC tau iris p53 complex exist in humans? And do these ARC tau EVs exist in humans? So I started with some co-immunoprecipitation in human postmortem tissue from Alzheimer's patients. So I got some tissue from control patients, Braak stage 2 and Braak stage 6 Alzheimer's patients. And then I immunoprecipitated tau from them. You can see the IgG samples are clean, and I was able to immunoprecipitate tau. When I blotted back for ARC in these samples, I was able to detect ARC and also iris p53. This shows that ARC tau and iris p53 complex is conserved in humans as well. Here I'm showing you the expression levels of ARC and tau in these postmortem human samples. Right here are control samples, and then right here are Braak stage 6 samples. I stained for 88, which is supposed to detect pathological tau. And we confirmed that we were only able to detect pathological tau in Braak stage 6 samples and not in control samples. I also blotted for tau 13, which looks at total tau levels. And you can see that the total tau levels were comparable between control and Braak stage 6 samples. And this is what I want you to remember, that all these samples do have comparable total tau levels. We also blotted for ARC and syntenin just to make sure that the levels were comparable between the control and Braak stage 6 samples. Now I used the exact same samples and isolated EVs from these human postmortem samples. Right here is an EM of a control EV and Braak stage 6 EV. So morphologically, they look pretty similar. We did not feel that there were any differences. When we did some NTA on these EVs, the median particle size was also comparable between control and Braak stage 6 EVs. So it seemed like there was not anything inherently different about the EVs that were isolated from control samples versus Braak stage 6 samples. I am showing you a blot here of the EV samples that were isolated from the same postmortem tissue samples that I showed in the previous slide. And then we blotted for 88, which shows pathological tau, and tau 13, which shows total tau. So in spite of having very comparable amounts of total tau, but different amounts of pathological tau, we saw that in the EVs, the amount of pathological tau that was getting packaged was different between control and Braak stage 6 samples. The tau 13 levels were a little bit more comparable, I would say. And in these same EVs, we were able to detect ARC. And I think what I want you to focus on is that the EVs that did have more pathological tau also had higher levels of ARC. That was pretty interesting. And right here, we have syntenin as positive EV marker. Since I was seeing this correlation by eye, I went ahead and quantified the amount of pathological tau and ARC, and then made a correlation plot for 88 expression and ARC expression in these EVs. And the Pearson correlation coefficient for this was 0.7, which is pretty high. So this shows that the levels of pathological tau and ARC that you can detect in human EVs is pretty highly correlated. So it is possible that ARC could be facilitating release of tau in human EVs as well. Next, we repeated double immunogold labeling in human EVs as well. And we saw a pretty similar pattern as we saw in the mouse EVs. There was a population of EVs that had just tau in them. We could also detect some EVs that had only ARC in them. And then there was a special population of EVs that had both ARC and tau in them. So it seems like all the effects that we did observe in mice was also present in human samples, which was great. So what we think might be happening is that in wild-type mice, when a donor neuron develops tau pathology, it has tau aggregates, which breaks down to make tau seeds. These tau seeds then bind to ARC, and ARC is already binding to IRSP53. So probably tau, ARC, and IRSP53 form a complex. And the tau seeds are then released in ARC extracellular vesicles. When these ARC extracellular vesicles are taken up by a recipient neuron, both ARC, tau, and tau seeds are released in these recipient neurons. And these tau seeds can then start tau pathology in a recipient neuron that's healthy. So overall, the tau accumulation and cell death would be lower in donor neurons in wild types. The tau release in EVs would be higher, and there would be more tau spread and seeding. When you knock out ARC in the donor neurons, the tau cannot be released in extracellular vesicles. So you see a lot more tau pathology in donor neurons. But only naked tau gets out of donor neurons. And so we see lower amount of tau spreading and seeding in recipient neurons in absence of ARC. But that also means that there is more tau accumulation and probably cell death in these donor neurons. So I hope I have convinced you today that ARC facilitates intercellular tau transfer, both in vitro and in vivo. ARC facilitates the release of tau in extracellular vesicles in mouse cortex. These ARC tau EVs can seed tau pathology. ARC directly binds with tau in GSD pull-down assays and co-IPs. Intracellular tau accumulates in neurons when ARC is not present. And ARC tau binding and release in EVs is conserved in humans. We are now planning to target ARC tau binding site and study its effect on tau release in EVs. We are also trying to see if ARC tau EVs are sufficient at seeding tau pathology in mouse brains. We are also looking into what are the levels of ARC tau EVs out of the total population of EVs that we see. And then we are looking into if tau binds ARC capsids specifically. With that, I would like to thank my lab. They have been instrumental in getting this work done. As you would have seen through my slides, a lot of people helped with so many different experiments. And with that, I'll take any questions.
Thank you very much, Mitali. It's really a great talk. We have several questions in the chat. So unless there are some that would like to ask a question, I will go through this question in the chat. So the the first question is from Philippe Askenazi, asking if there might be ARC EVs in the CSF. And whether there might be a role for ARC in the immune system.
That is a great question. Yeah, it's possible that ARC EVs are also present in the CSF. We have tried isolating extracellular vesicles from CSF, but as probably you guys know, that the yield is really low and ARC EV population itself is low. So we have to optimize that a little bit more. As per my knowledge, ARC is not known to play a role in the immune system.
Actually, it is. So there's, I mean, we don't know for sure exactly what's happening, but ARC is expressed in a particular kind of dendritic immune cell. But there's only one paper on this. And they did look at the ARC knockouts and they saw some immune phenotypes. In my lab, we haven't followed up on it directly. But yeah, there is a possibility that ARC is involved in the immune system. It's mostly almost exclusively expressed in the brain otherwise.
Yeah, but it is possible that these tau-containing ARC EVs are taken up by microglia and then can induce an immune response. That is very much possible.
Okay. And then there is another question, but I guess that you partially answered the question, but I will repeat the question again anyway, which is about the phosphorylation state of tau before and after the transfer of EVs. Maybe you can
Yeah, I mean, in our transfer
the model that you have.
Yeah. In our tau transfer assay, we just looked at total amount of tau and we did not specifically look at phosphorylated tau. So we cannot say confidently that it's the phosphorylated tau that's getting transferred. That being said, the EVs do contain phosphorylated tau, does seem to have a higher binding affinity for hyperphosphorylated tau, and they can see tau pathology. So, yeah.
And Brad Hyman's group has shown in their publications that transfer assay is somewhat agnostic to the state of tau. So you can still see transfer of monomeric tau, oligomeric tau, hyperphosphorated tau. So it's sort of a global sort of transmission assay. It's not specific to pathological tau, although of course, we think that a component of this is certainly capable of spreading pathological tau.
Another question from Philippe Askenaze. Do microglia or macrophages play a role in ARC traffic?
Yeah, that is a little bit complicated. Neurons express much more ARC, obviously. It is possible that microglia can take up ARC EVs and then maybe make their own EVs that could be involved in ARC and tau trafficking or spreading. But I don't think the levels of ARC in microglia inherently are very high.
This is something we're going to look at. We've developed a tool to try to look at ARC transmission in vivo rather than culture. And so in our culture system, we don't have a ton of glia and we don't have any microglia. So of course, we're mostly looking at cell-to-cell transmission in that context. But in vivo, of course, it's totally possible that there's some of the EVs are being released via microglia, and we can't, tease them out. So we've built a tool that we're going to do look some experiments to see if we can detect transfer directly of ARC from neurons to microglia.
Okay. There is another question from Christina Kuglan. Can you share how you determine ARC forms or may form a capsid compared to a protein corona?
I'm sorry, yes, that's the question. I just wanted to know how you determined it was a capsid versus a protein corona.
So the history of this is that we weren't looking for this at all. So we were trying to purify ARC protein from E. coli just to do some biochemistry. And we kept on noticing that the protein was crashing out of the size exclusion column as if it was much bigger than it should be. And when we put that protein that we had collected off the exclusion column on a grid for EM, that's when we noticed that it forms these capsids. So the protein itself is capable of forming the capsid independent of anything else. It's a spontaneous assembly process that's similar to most retroviruses. And so all the experiments we've done, from like Mitali showed, in vivo collection of EVs, etc., ARC is always protected inside the membrane. So any protease protection assay, the ARC is not on the outside, so it's not the corona.
Awesome. Thank you.
Maybe someone wanted to make a comment about this question. Saker?
No, actually, I have another question. So.
Oh, okay. So go ahead with your question.
So my question is, have you, observed any alteration in the conformation or phosphorylation state of the tau itself? Like, does ARC alter the conformation or phosphorylation, or just it's a mechanism of transmission?
Great question. Thank you. Yeah, we have not looked deeply into it, but we did do some experiments where we basically incubated either wild type ARC or capsid ARC with purified tau and then let it incubate for 24 hours and then put them on an EM grid to look at if there were any changes. Yeah, very preliminarily, we could not see any differences. That being said, we have not looked any deeper into it.
So it's an interesting question because we, and this is again sort of not something that we've delved into deeply yet, but when we express both ARC and tau in cell lines, for example, they form condensates, and we're trying to figure out in that condensate what the status of tau and ARC are, and there's definitely synergistic effects where, ARC could be affecting the oligomerization state of tau and vice versa. We're collaborating with a group to do some biophysical modeling of that interaction, but it's not a straightforward interaction for sure. And there's actually multiple context sites that tau and ARC make together. So it's going to be interesting to tease out the sort of interaction there.
Thank you, Jason. Thank you, Mitali. That's a great work. Thank you.
Thank you.
Okay, I guess there are also some questions about the molecular composition of the ARC ARC positive small EVs. I had one about this, and also there is one about this question from Alessandro Scacchetti, I guess, specifically about whether there might be some RNA inside this ARC positive and tau positive EVs, and could the tau-ARC interaction be mediated possibly by RNAs or nucleic acids?
Yeah, those are all great questions. So to begin with, I am quantifying the amount of ARC tau EVs right now. And one of the problems that we have run into is when we do double immunogold labeling, basically the 15 nanometer gold particle has a harder time getting inside the EV. So that kind of like low balls the amount of ARC tau EVs. But what I've seen is the amount of ARC EVs are around about 10%, and then ARC tau EVs are probably around about 7-8% so far from my quantification. And then somebody asked, is there RNA inside ARC tau EVs? I have not specifically looked at ARC tau EVs and tested for RNA in them, but we do know that RNA is present in ARC EVs, so it's highly probable that is the case. There are other people in the lab that are looking into it.
Yeah, and so we, to the question of sort of, have we globally figured out what's in ARC EVs, and that's sort of been more difficult than we expected, partly because of this issue of being a subset of total neuronal EVs. So we've done some unbiased proteomics and RNA sequencing, but we still sort of trying to sort out what that data actually means in terms of validating them. So we don't have an answer yet for sort of here's a set of proteins and RNAs that are in there. We know for sure that ARC encapsulates its own mRNA, but this sort of synergistic effect of nucleic acid on tau, of course, is also interesting in that tau is an RNA binding protein, and that's another sort of biochemical avenue, that we're sort of investigating, but nothing concrete yet.
Mm-hmm. I guess we have to move on with all the other questions today. There is one about from Frédéric Naftolin saying that estradiol blocks tau phosphorylation, and does sex affect the tau ARC system?
Yeah, so that's a great question. There's definitely some sex-specific effect. So the tau transgenic mice, even in humans, we know that females have much worse tau pathology in comparison to males, and that is definitely recapitulated in these tau transgenic mice. The females do have much more tau in them. That being said, yeah, I mean, we have not seen sex-specific effects in the packaging of tau in the EVs, basically.
Another question from Frédéric. Is there tubulin and or actin in the ARC tau EVs?
That is a great question. Another grad student in the lab has been trying to characterize ARC EVs really well. So they're looking into if, tubulin, other different proteins, cytoskeletal proteins are present in the ARC EVs. I have not looked at that for ARC tau EVs specifically. Yeah.
Yeah, and we are quite interested to know. I mean, so we think, of course, that ARC is normally involved in EV release and intercellular signaling, and that's another whole body of work that there's a preprint out on in its role in synaptic function and plasticity. And so this disease context could alter for sure the composition of ARC EVs, both because of the tau that's coming in as well as whatever else is being packaged under those abnormal situations. So that's, we definitely want to get a handle on that.
There are other questions. I'm trying to see whether they may focus on the same points. Let me see.
Yeah, I mean, some of these so Natalie brought up a good point where, and we've talked to Stephanie Fowler, who's author, first author of that paper. And so she sees tau in multiple EV populations. The cryo that they observed, these filaments are for sure in these rare large EVs. And we actually, I mean, we didn't, a lot of our experiments, we discarded the large EV fraction because we were concentrating on these small EVs. So we for sure don't think that ARC is the only way to generate EVs that have tau. And I also think that the composition of the tau in these EVs is going to be different depending on what stage of disease you're looking at. And our hypothesis is actually that ARC is involved in tau transmission very early in the disease, partly because in Alzheimer's especially, you get this hyper neuronal activity phenotype where inhibitory cells are lost first, and so the excitatory neurons are hyperexcitable. You get more ARC expression, and that contributes to that transfer of tau early. But as you get to the point where cells are dying and there's filament, neurofilaments, we think then that ARC is actually decreased.
Do you have an idea? I mean, could you share your prospective views on what could be the role of these ARC small EVs in normal conditions, and whether the interaction between normal tau and ARC in normal condition could basically be interesting to understand?
Yeah, I mean, Mitali showed that even in control human tissue, she can observe an ARC tau interaction, and there's been some papers showing that tau itself may actually be involved in synaptic plasticity and memory under normal conditions, and so that's a question that we're sort of trying to address as well, which is this a pathway that's just hijacked during the disease? Or is it a natural pathway? And if it's a natural pathway, I don't really have a good answer for why you would want to transfer tau itself in terms of, plasticity and function. But that's something we could we're interested in looking at.
Natalie Turner would like to ask a question. Natalie?
Yeah, hi, Jason. Thank you so much for answering my question. I guess I was very interested in the differences because there's so much interest in tau packaging and how it may be involved in disease progression via EVs or other mechanisms. And now there's been more studies looking at these larger EVs like amphicectosomes, amphisomes, some really recent work on those. And also what we know about Alzheimer's as it could have this lysosomal disease component as well. And there seems to be a lot of converging hypotheses. And yeah, I guess that was kind of what I was getting at. It's like the ARC could definitely be one way, and it may be constitutive in normal states, it may be pathological, but then you could get this influx of other biogenesis pathways that are being contributed by other subcellular organelles or biogenesis pathways. So do you have plans to follow up in other directions, or are you just going to stick to ARC? Are you going to start looking at these other EV populations? Like, what's the plan, and have you thought about those kinds of mechanisms?
Yeah, I mean, I think we were surprised by the phenotype initially, that it was so strong when you do this tau transmission assay, where it really did look like under those conditions, ARC was important, almost essential, because there was very little transmission. And I think that, but as you say, like the next stage of this is really more in looking at the seeding capacity of these EVs. If you inject them into an older animal, can they then induce spread? Doing these causal experiments, of course, in human tissues you can't do it. So I think we're going to have to figure out in the mouse models what we're seeing. But I think your point about the different forms of EV tau is certainly a good one. I mean, I think there's also the microglia component, the astrocyte component. There's clear evidence that that they're transmitting tau in some way, and they can spit out their own EVs. So those EVs, so the brain EVs, of course, that Stephanie characterized, you can't tell for sure whether they came from neurons or not, because again, they're sort of they're just brain isolation protocols. But in our case, when we do this in cultured neurons, we know for sure that they're coming from neurons because we don't have any glia in those cultures. So, I think there's a lot to tease out there, and we're certainly not saying that ARC is the only player in this at all.
It would be interesting, I guess, that to look at the effect of tau, phospho-tau transferred by ARC EVs to naive neurons that never saw phospho-tau, and see whether that could immediately induce some sort of dysfunction in terms of immediate early response, or synchronization, using some sort of device where you can look at neuronal connections.
Right. Yeah, no, I mean, this is always the exciting part of stumbling onto new science, is that there's lots of questions to follow up on. And certainly like what the tau EVs, the ARC tau EVs are doing. So we've got another paper where if you just put in EVs with ARC in them, they can transfer ARC into neurons and affect synapses. And we can do a similar experiment when we take out ARC tau EVs and see what's happening.
Right. All right, so I guess that we have to conclude this EV Club. We had so many questions today, so thank you so much, Mitali and Jason, for sharing your data and your views and responding to and answering all these questions.
Yeah, no, thanks everyone for their questions. Those are all good ones.
Yeah, that's great. And do not miss the next webinars from EViNS on the in the EV Club series. And thank you so much for being here today, listening to Jason and Mitali about tau, EVs, and Alzheimer's disease. Ken, would you like to conclude this EV Club?
You said it all. So thank you so much. And we hope to see everybody again sometime soon.